The overall goal of this procedure is to characterize the composition of extracellular matrices from tissues by mass spectrometry. This is accomplished by first disrupting the selected tissue until it is completely homogenized. The second step involves decellularizing the tissue to enrich for extracellular matrix proteins while concomitantly depleting any intracellular components. Next, the ECM-enriched protein sample is denatured. In the final step, the denatured ECM-enriched protein sample is deglycosylated and digested into peptides. Western blot analysis is used to monitor the quality of the decellularization and ECM protein enrichment. Ultimately, analysis by mass spectrometry of the peptides permits characterization of the composition of the ECM for that particular tissue.
This method can provide insight into the composition of the extracellular matrix of normal tissues, but can also be applied to other systems, such as diseased tissue like cancer.
To begin, prepare all the buffers as indicated in Text Protocol Table 1, including the addition of protease inhibitors. Then, disrupt 100 mg of tissue in 500 microliters of buffer C with a tissue homogenizer until a uniform suspension is obtained. Transfer the homogenate to a tube rotator and incubate for 20 minutes at 4 degrees Celsius. Pipette 10 to 20 microliters of the sample into a tube labeled as total tissue extract or starting material. Flash freeze the sample in liquid nitrogen and store it at -80 degrees Celsius. Next, centrifuge the homogenate at 16,000 times G for 20 minutes at 4 degrees Celsius. Collect the supernatant in a clean tube and label it